Have you ever stopped to think about what's actually going on inside of you, like at a totally microscopic level? Well, today, we're diving in. We're going to take an incredible tour of the bustling city that is the eukaryotic cell. That's the basic building block for everything from plants to people. And we're going to explore the amazing little machines, the organelles, that keep the whole show running.
But first, just picture this for a second. What if you could have an entire, fully functioning city, I'm talking power plants, factories, transit systems, the whole deal, and have it all exist in a space so tiny you can't even see it?
You know, that's not science fiction. It's reality. Every single one of your cells is that complex. It's a miniature metropolis. So in this explainer, we're going to be the tourists, checking out the essential infrastructure that makes life itself possible.
All right, our first stop is the city's power grid. This is the tireless generator that fuels absolutely everything else. We are, of course, talking about the mitochondria. Seriously, without these things, the entire cellular city would just go dark. So check this out. This diagram gives us a rare glimpse inside a mitochondrion. They're usually so small they're invisible under a regular microscope unless you use special stains. And you can see right away what makes it so special. This complex inner structure with that outer membrane and then this heavily folded inner one. That design creates these separate compartments, which is totally key to how it works. And yeah, we often draw them as these little sausage shapes, but what's really cool is how their numbers change. A cell that needs a ton of energy, like one of your muscle cells, is going to be absolutely packed with thousands of them. A less active cell? Way fewer. It's a perfect example of form following function in biology. And to give you a sense of scale here, which is just mind-boggling, the average diameter of one of these is between 0.2 and 1.0 micrometers. A micrometer is 1/1,000,000th of a meter. We are truly in a miniature world, where every last bit of space is used with incredible efficiency. And they're typically about 1 to 4 micrometers long. So they're super compact, incredibly powerful, and just perfectly built for their job as the cell's energy producers. You know, the mitochondrion's whole structure is the secret to its power. It's got this really clever double membrane design. There's a smooth outer membrane and that super folded inner membrane. This setup creates two totally separate spaces, which is absolutely critical for how it generates energy. And this right here, this is one of biology's most elegant engineering tricks. That inner membrane is folded into these complex loops called crista. So why do that? It's all about maximizing surface area. Think about the fins on a car radiator. They're there to get rid of heat more efficiently. The crista do the same thing, but for creating energy. More surface area means more space for all the chemical reactions that power the cell. It's genius. And deep inside that inner membrane, you find the matrix. Now, this isn't just empty goo, it's a bustling chemical workshop. And here's a super fascinating clue we're going to come back to later. The matrix actually has its own circular DNA and its own little protein-making factories, a specific kind called 70S ribosomes. It's almost like a little semi-independent creature living inside the cell. So all of this complex machinery, all these folds and compartments, it's all for one main reason: aerobic respiration. That's the process that takes the food we eat and turns it into the cell's universal energy currency, a molecule called ATP. And this, right here, is exactly why you've always heard mitochondria called the powerhouses of the cell. Okay, so our city has power. Now it needs to produce goods and food, right? Well, in plant cells, and in some cool organisms called euglenoids, that job falls to another set of organelles, the plastids. Think of these as the city's factories and farms, responsible for making and storing all the vital materials. Plastids are basically sorted by the kind of pigments they have inside. You've got the famous chloroplasts, which are packed with green chlorophyll to do photosynthesis. Then you have chromoplasts, which hold the pigments that make fruits and flowers that vibrant yellow, orange, or red. And finally, you've got leucoplasts, which are colorless and basically act as little warehouses for storing stuff like starch and oils. So let's zoom in on the most famous plastid of them all, the chloroplast. As you can see in this diagram, just like the mitochondrion, it's got a complex internal design with an inner and an outer membrane. But inside, whoa, it's filled with these incredible stacks of membranes. This is the green engine that powers pretty much the entire plant world. Inside the chloroplast, you find these organized, flattened sacs called thylakoids. The best way to think of them is as the cell's solar panels. They're often arranged in these neat piles that look like stacks of coins, and those stacks are called grana. The thylakoid membranes are the exact spot where that light-trapping chlorophyll is located. Now, the fluid that surrounds all these stacks is called the stroma. And a lot like the mitochondrial matrix, the stroma is a super busy place. It's got enzymes for building carbohydrates, and hey, sound familiar? It also has its own small circular DNA molecule and those 70S ribosomes. Okay, so our city has power from mitochondria and food from chloroplasts. Now it needs workers to actually build things, everything from enzymes to the structural beams of the cell. Let's meet the ribosomes, the tiny but incredibly numerous protein factories of the cell. Now here's a really key detail. Size matters. Out in the main city, the cytoplasm, you find bigger ribosomes called 80S. But inside our power plants and farms, the mitochondria and chloroplasts, you find a smaller 70S type. And that S just stands for Svedberg unit, which is a measure of how fast something sinks in a centrifuge. It's about density, not simple math, which is why the subunit numbers, like 60 and 40, don't seem to add up to 80. A little weird, I know. These little guys were first spotted by George Palade way back in 1953. And even though they're tiny, these granular structures are absolutely essential. Every single protein the cell needs to function, to grow, to do anything, is assembled right here by the ribosomes. All right, our final stop on this tour is the city's internal framework. This is its highway system, its support beams, and its communication network, all rolled into one. This is the cytoskeleton, an incredibly intricate and dynamic network of protein filaments. Seriously, without the cytoskeleton, a cell would just be a formless, mushy blob. It provides the mechanical support that gives a cell its unique shape, whether that's a long, stringy nerve cell or a flat skin cell. But it's not like static scaffolding. It's constantly changing, breaking down and rebuilding, which allows the cell to move, to divide, and to shuttle materials around inside. It's pretty amazing. So as we kind of zoom back out, let's just do a quick recap of the key pieces of machinery we've seen on our tour of this incredible cellular city. We saw the mitochondria, using those folded cristae to create a huge surface area for making ATP. We explored the chloroplast, with its stacked thylakoids perfectly designed to trap sunlight. And we met the ribosome, the simple two-part factory that's essential for building every protein. Each one is just a masterpiece of design and function. And that leaves us with one last and really fascinating question. Remember that weird clue we kept finding? The fact that both mitochondria and chloroplasts have their own circular DNA and their own 70S ribosomes, the exact same kind you find in modern-day bacteria? Why would these organelles living inside our cells have their own separate genetic system? What could that possibly tell us about where they came from, deep in their evolutionary past? It's definitely something to think about.